Welcome to Anatomy 101.
Muscles. Our muscles allow us to move our bodies. Even when we sit perfectly still, muscles throughout the body are constantly moving.
Our muscles are very important. We use them every single day.
We use them so much, even during simple activities such as lifting a box, eating food, and even lifting a friend. Our skeletal muscles, which are voluntary muscles, produce movement by pulling bones as they contract in different ways. Each skeletal muscle, such as our rectus abdominis, your quadriceps, your biceps brachii, and more, are made up mostly of muscle tissue, nerve fibers, and connective tissues that support and tie the bones and the muscles together. Isn't that amazing?
Can we please get to the good part?
The internal structure of a skeletal muscle contains lots and lots of layers. It demonstrates a bundle within a bundle structure. They are divided into 5 groups. The myofibrils, which are thousands of thread-like structures compressed together to form muscle fibers. Muscle fibers, also known as your muscle cells, are just like the common cells with mitochondria, nuclei, and sarcolemma, which is a plasma membrane. And these muscle fibers form larger rope-like bundles called the fascicles, which then squish together to form an entire muscle organ. The tiny myofibrils that bind up to form your muscle fibers are composed of individual contractile proteins called the myofilaments, which then consist of thick filaments and thin filaments. Myofibrils are composed of repeating sections of a sarcomere, which appear under the microscope as alternating dark and light bands. Now, let's start off by asking, what is a sarcomere? A sarcomere is the smallest contractile unit of muscle and is the functional unit of a myofibril. Note that the striated appearance of the sarcomere is due to the arrangement of the thick and thin myofilaments within each sarcomere. But what is a sarcomere made of? A sarcomere contains both thin filaments called actin, which are made up mostly of 2 twisty and light actic strands and thick filaments, and myosin, which are composed of thicker myosin strands. Actin, the thin filament, consists of actin molecules that form an actin filament and are twisted into a helix. Each actin molecule is a myosin binding site where the myosin head can attach. But wait, there's more. Tropomyosin covers the myosin binding sites on actin, which blocks myosin from binding where the muscle is relaxed. Troponin holds tropomyosin in place and changes shape when calcium ions bond. Troponin also moves tropomyosin away from myosin binding sites on actin, and then muscle contraction begins as myosin can bind to actin. Myosin is a motor protein that can convert chemical energy in ATP to the mechanical energy of motion, aka the production of force. But what does myosin look like? Myosin consists of a tail and a head, with the myosin head being the actin binding site, an ATP binding site which hydrolyzes ATP to generate energy for muscle contraction. Now, here's some fun facts. Each myosin can interact with 6 actin, and each actin can interact with 3 myosins. Wow, the power of science! Wow! Each sarcomere is segregated by a line called the Z line, which is located at either end. This acts as a border formed, and it alternates the thin filaments in a kind of zigzag pattern. Now, have you ever gone to the mall looking for a very specific shirt with a very specific pattern that you like? Well, sarcomeres also have components that contribute to the banding pattern of the sarcomere. There is an I band, which is also known as the light zone, and this contains light filaments so light can get through. There's also an A band, which is also known as the dark zone, where the light doesn't travel through very well, hence the dark appearance. In addition, there is a M line, found right in the center, which wraps myosin filaments to keep them bundled together. Sounds pretty cozy, right? Finally, there is a titin. Drumroll, please. The titin is one of the biggest proteins in the body, and it spans from one end of the sarcomere to the other end. Just remember that muscles contracting is all about sarcomeres contracting. When a sarcomere contracts, the Z discs are brought closer together. When your muscle fibers are in the relaxing state, the muscle's actin and myosin strands don't touch, even though they really want to. But there is a theory that can make this possible. The sliding filament theory, also known as the cross-bridge theory, describes how the 2 ends of a sarcomere come closer and how the actin slides past the myosin so they could overlap more during contraction. It's a pretty complex process, and that's why we are here to help you understand.
What is the
this mean? I don't understand this.
Let's say you're studying for an exam, an anatomy exam. You're really having trouble understanding this whole sliding filament mechanism thing, and you have all your papers splayed out on your desk. You start putting things away and you're about to give up when you slide one scattered paper over another. That's it. It finally clicked. And you are about to understand the sliding filament theory of contraction, my friend.
The sliding filament mechanism of a contraction, also known as the sliding filament theory of contraction, is when a muscle contracts and the thin filaments of a sarcomere, actin, slide over thick filaments, myosin.
When we see pictures of contracted sarcomeres, we can see that the I band or the light zone of the sarcomere is decreased in size. This is because the I band represents the thin filaments with no overlapping with thick filaments. During a muscle contraction, the thin filaments will be pulled towards the center of the sarcomere, thereby causing the thin filaments to overlap with the thick filaments, causing the Z discs to be closer in distance and the I band to be shorter. It's important to know that just like how in our sliding papers example, none of the papers actually changed length, they simply overlapped. It's the same thing with sarcomeres. The actual lengths of the thin and thick filaments never change, they only overlap, which causes the I band, also known as the light area of the sarcomere, to decrease in size.
Did you get it?
But how are thin filaments slide over the thick filaments? I don't understand!
Let's first talk about the thick filaments, myosin. A thick filament is made up of end-to-end myosin molecules. Myosin is able to pull actin towards the center of a sarcomere and therefore increase overlapping by binding to actin using the actin binding sites present on the globular head of the myosin molecule.
The myosin head also contains a myosin ATPase, which will break down ATP, giving myosin the energy to perform a physical movement called the power stroke.
But what about the thin filament?
Good question. The thin filament is actin molecules that form two coil chains. The myosin heads bind to the thin filament at the cross-bridge binding site on the thin filament. However, see, the cross-bridge binding sites on actin are blocked by these molecules called tropomyosin, which run along actin. Another molecule called troponin holds these tropomyosin molecules in place.
So how do we get these tropomyosin molecules out of the way so my muscle can contract?
Never fear, calcium is here!
Whenever our muscles produce movement, what happens first is our brains send an action potential in the form of an impulse along the motor neuron, which is a nerve that connects to the muscle. Calcium channels, which are ligand-gated ion channels, open in the sarcoplasmic reticulum when the motor neuron releases a neurotransmitter, acetylcholine, into the synapse. The acetylcholine binds to the receptor sites of ligand-gated channels, causing the muscle action potential to activate. The action potential then travels down the T-tubules, tubes that are located in the sarcolemma, the plasma membrane of the muscle cells, which causes a change in the voltage-gated calcium channels. In this case, these calcium channels release calcium ions into the sarcoplasm while they are open. Calcium binds to troponin and changes its configuration, which pulls tropomyosin away from the cross-bridge binding site and allows for actin-myosin interaction to occur. When calcium is removed, tropomyosin moves back into place, blocking the cross-bridge binding site once again.
That kind of sounds like it's a cycle.
That's because it is. The whole cycle of contracting a muscle is called, well, this contraction cycle. It's also known as the cross-bridge cycling.
The cycle can begin anywhere, really, but let's start at when the myosin heads break down ATP and become reoriented and energized. Myosin in this stage of the cycle is pulled back and ready to bind to actin. The myosin heads then bind to actin, forming cross bridges.
These myosin cross bridges rotate towards the center of the sarcomeres, a movement known as the power stroke. As myosin heads bind to a new ATP molecule, the cross bridges detach from actin, and myosin and actin separate.
Oh, so that thing known as rigor mortis can be explained by the contraction cycle, because when a person is dead, there's no more ATP being generated, and so the cross bridges will remain attached, leaving the muscles contracted and stiffed until the body's proteins are naturally eaten away by enzymes released by cells after a person dies.
So, to summarize, the sliding filament theory of contraction is when a muscle contracts because thin filaments, actin, slide over thick filaments, myosin. The I-band or light zone of a sarcomere decreases in size during a contraction of a sarcomere because of the increase in overlap of thin filaments and thick filaments.
But the actual length of the thin and thick filaments never change. Myosin is able to pull actin towards the center of a sarcomere using the actin binding sites present on the globular heads of the myosin molecule, but only when calcium changes the configuration of troponin, which moves tropomyosin out of the way of the cross-bridge binding site on actin.
The whole cycle of contracting a muscle was called, what was it again? Oh yes, the contraction cycle. And the contraction cycle requires ATP.
Some other cool fun facts are there's an optimum amount of overlap between actin and myosin, which will allow the most shortening of a sarcomere possible. Fewer myosin molecules can interact with actin molecules when muscles are overstretched because they're pulled apart. When muscles are understretched, no more shortening can occur because thin filaments are already too close. Shortening of sarcomeres causes shortening of the whole muscle fiber, which causes the entire muscle to shorten.
Wow!
Our muscles surely do a lot of work. But ever wonder about how our muscles get bigger when we go to the gym, exercise, or even just use them to do some work? It's definitely not magic. It's just muscle hypertrophy. When we consistently challenge our muscles to deal with higher amounts of resistance or weight, our muscular size grows. But the question is, how do our skeletal muscle fibers increase in size? And what makes our muscles grow? Now, let's dig a little deeper, shall we?
Most cells in our body undergo a process called cell division for growth and repair. However, there is a different case for skeletal muscle cells. Once a single skeletal muscle starts to mature, the muscle fibers are also starting to lose their ability to undergo cell division. So instead, the growth of a skeletal muscle mainly relies on hypertrophy.
By definition, hypertrophy is the enlargement of a muscle caused by an increase in the size of its cells. But wait, this is not to be confused with hyperplasia, which is a whole different story. Hyperplasia is a process in which the number of the muscle fibers increase within a muscle, while hypertrophy is the process in which the size of the muscle fibers increase within a muscle.
By using your muscles to do work, such as engaging yourself to do a forceful, repetitive muscular activity or training, you are stimulating the production of actin and myosin filaments, thus stimulating a continuous production of ATP as we learned in the previous episode. This is when your muscle fibers thicken, become larger and stronger, which is needed to handle the increased load. As a result, a cellular response takes place to generate more components.
For our muscles to grow, stimulation and regeneration must occur. This is where the role of satellite cells, dormant cells that reside between the basal lamina and sarcolemma, becomes important.
The satellite cells become activated by muscle trauma, damage, or injury. These cells facilitate muscle hypertrophy by dividing slowly and retaining their ability to fuse with one another or with damaged muscle fibers to regenerate functional muscle fibers. To successfully release growth factors, hormonal responses are needed. Hormones, such as testosterone, promote further enlargement of muscle fibers and thus help regulate cell activity.
Satellite cells proliferate to the damaged tissue as a result of this process. They bind to the skeletal muscle and transfer their nuclei to the muscle fibers, allowing them to thicken and grow in size. So what is the lesson here? Our muscles rely on more than just activity to grow. Having proper nutrition, hormones, or even rest is still needed because without these things, our bodies might not be able to repair muscle fibers that have been damaged.
This is why it is important for us to use our muscles, whether in doing simple activities or any other physical activity really every day. Sometimes it's okay to be a couch potato, but if we do not continuously expose our muscles to some resistance, our muscles will shrink. And yes, there is also a term for this, and it is called atrophy. This is when your myofibrils start to break down and are not being replaced. As a result, there will be fewer myofibrils in the muscles, thus decreasing muscle size.
Wow, that was a lot of interesting information. We learned that muscles are made up of many different components such as sarcomeres that are then broken down into even more components. Muscles contract by using cycles that involve sliding thin filaments over thick filaments. And lastly, our skeletal muscles can increase or decrease depending on how we use them and other factors. With muscles, there's always something cool to learn. Now go put your body to work.
J. Rest.